Okay, the third topic that is needed to be discussed under the chemistry of life is about the DNA structure. We all know that DNA is one of the forms of nucleic acids, so we can include or we can give an introduction on what nucleic acids are. So, introduction. Okay. Because if we directly discuss about the DNA structures, then there is a possibility that the students might be surprised because they don't have like any background in chemistry. So it's best to like walk them through this process. So introduction. So in the case of nucleic acids, we have to elaborate as well. We have to explain that nucleic acids are one of the biomolecules that are known as the information molecules. So instead of getting it from other sources, unlike carbohydrates, lipids, and proteins, these nucleic acids are biomolecules that are built together to become information molecules, to keep information. Okay, so right after the introduction about nucleic acids, then we can continue with the nucleotide structure. Okay, so nucleotide structure, this is the building block of nucleic acid. So by knowing the very simple or the most fundamental part of what a nucleic acid is, then we can build the DNA structure from there. Okay, so we have under the nucleotide structure, we have to include the components. Okay, components such as the pentose sugar, which is discussed in the previous subtopic. Okay. The phosphate group, and the third component, which is the nitrogenous bases. Okay. If a diagram is going to be provided for the student, it is also better to provide a diagram or an example of a chemical structure for each component, because we want to show to the student that the process of how we are going to build the nucleotide molecule or the nucleotide unit. So by showing the chemical structures together and incorporating the chemical reaction, condensation reaction, then the student would understand that the nucleotide unit is going to be built in this process. Okay, so under the nitrogenous bases, the types of the nitrogenous bases has to be, the student has to be familiar with the types as well. Okay, for example, we have the purine and the pyrimidine. Okay. But the good thing here is that students doesn't need to memorize the exact structure of what purines and pyrimidines are. You can just say that purine is a single ring nitrogenous base, whereas for pyrimidines they are double ring. So if a DNA double helix is going to be formed together, this is what's going to look like. But the students are not they are not really required to know the exact structure of what this nitrogenous bases are. Okay. Okay. So right after the familiarization in the nitrogenous bases, we can now form, or we can now add more nucleotides together to form polynucleotides. Okay, so the formation of a polynucleotide. Okay, so when you say polynucleotide, you condense all the nucleotide units together in a single strand. So in a polynucleotide, the things that are needed to be elaborated on are the chemical reactions that are involved in the process. So this includes condensation reaction, okay, and the bonds that are formed. Okay. So is it going to be covalent bonds? So for example, if the components under the nucleotide components are going to be formed together, pentose sugar phosphate group and nitrogenous bases is going to form covalent bond. But if we are going to condense nucleotide units together, it's going to form a different bond, which is what we call a phosphodiester bond. In SL students, phosphodiester bond can also be called as covalent bond, okay? Because they don't need to learn as much much complicated terms. So phosphodiester bond is only applicable in HL students. Okay. So once we have formed the polynucleotide, we can now form the structure of the DNA, which is the double helix. Okay, or show the students the double helix structure. Okay, so you have to elaborate as well that the DNA double helix is formed out of two polynucleotides together, wherein the nitrogenous bases between the polynucleotides are bonded together using hydrogen bonds. So it depends on the bases that are involved together. So the DNA double helix structure is achieved because of complementary base pairing. So you can also tell to the students that complementary base pairing is also a way of checking whether correct bases are being attached to the parent strand. Okay. So in this way, mutations are avoided. Complementary base pairing. Okay, so by knowing the informing about the complementary base pairing, you can also tell them that the adenine and thymine are bonded together using 2 hydrogen bonds, while cytosine and guanine are bonded together using 3 hydrogen bonds. So, okay. So for example, if there are in exams, if there are any structure-based questions, then we might be able to, the students will have a time to pinpoint which nitrogenous bases are present in that question. Okay, so another thing, okay, that is needed to be remembered, that DNA double helix, under the DNA double helix structure, they have to be informed about the bonds that are formed within the whole structure. Okay, so to cap off everything, the whole process of the DNA structure, that the formation of the DNA double helix is actually a semi-conservative process, meaning the original DNA is going to be used as a template to form the new DNA.